So first of all, let me thank the IMP as well as the Max Birnstiel Foundation for recognizing our work. And it's an honor for me to be here today and share this work. So my name is Sam. I'm finishing now my doctoral research at the Technical University of Delft, which is a city in the Netherlands that is now taking on these nice, vivid autumn colors, just like Vienna is doing. And it is here that we discovered this new, quite peculiar player in the CRISPR universe, namely an RNA-regulated CRISPR protease. And this story started about 3.5 billion years ago, which is not when my PhD started, although it sometimes feels like that. No, this is when it's approximated that the ancient interaction between viruses and bacteria started. So a typical interaction looks like this. So the virus attaches to the membrane of the bacterium and injects its DNA. Then it will hijack the transcription and translation machinery to make more of itself, to the point that the bacterium bursts open and these viruses escape and continue their hunt. This is obviously a detrimental outcome for the bacterium. So during the course of evolution, the bacteria have evolved really a plethora of immune systems, and one of the most famous ones being the CRISPR-Cas immunity. So in a nutshell, the CRISPR-Cas systems are typified by the presence of a CRISPR array, which is basically a memory bank that contains snippets of viral genomes that the cell has previously encountered. Next to that are the Cas genes, so that upon expression of this system, the CRISPR array is processed into guide RNAs, which can form complexes with the Cas proteins. And then, depending on the nature of the Cas genes, so if these are RNases, the guide RNA will guide them to the transcripts of viruses, and when it's complementary, the Cas enzyme will then cleave those transcripts to abrogate the viral infection. So in early 2020, a new CRISPR-Cas type was predicted bioinformatically to occur in primarily marine bacteria. And it was fairly large, 1,700 amino acids, and it was termed G-ramp, and the G also stands for giant. And if you look at its domain architecture, you can see the presence of these two RNases there. Also this peculiar TPR-chat protein, on that more later. So upon seeing this, we wondered, is G-ramp an RNA-targeting CRISPR-Cas enzyme? And in order to figure that out, we transplanted the whole system into E. coli, because you cannot really work with these exotic bugs. And then did biochemical characterizations, and then we indeed found that G-ramp is a giant protein that forms a complex with the guide RNA, such that it can sequence-specifically cut the target RNAs in two precise locations. You can also program G-ramp with the guide of your choice, so then you can also cleave a target RNA of choice, which could be beneficial if you want to do RNA knockdown or RNA editing. But this was really the point that the story got very interesting, because, as I said, this weird TPR-chat protein, also unknown at the time, is often co-occurring with G-ramp, and we wondered what that was doing. So domain analysis showed that it's a two-domain protein with a chat domain C-terminal, which is a Craspase-like protease, typified by the presence of this histidine and cysteine residues in the catalytic pocket. And then N-terminally, you have the TPR domains, which is basically a bundle of alpha helices that is often involved in protein-protein interactions. So given this fact, we wondered, do the G-ramp and the TPR-chat proteins also physically interact? In order to study that, we performed co-purification assays, and then we find indeed that if we purify G-ramp in the presence of TPR-chat, that would pull along in a very stable fashion. They were really glued to one another. And this was later also confirmed by the cryo-electron microscopy structures that we solved. As you can see here in yellow is TPR-chat docked on top of G-ramp. And then with the guide RNA here threaded nicely through the whole complex. And at this point, it started occurring to us that Craspase, as we now termed it, standing for CRISPR-guided Caspase, that this is an RNA-regulated protease. That binding of this RNA target to the guide RNA is somehow switching on the TPR-chat protease, and then upon cleavage of that target RNA, that the protease is switched off again. But in order to test that hypothesis, we needed to know what the target protein is, right? So I tried a whole bunch of proteins, and then it turned out to be hiding in plain sight, actually right next to GRAMP, is this protein called CSX30, which is a protein of unknown function, but when we incubated it together with Craspase, in the presence of a non-target RNA, so a target RNA that's not matching to the guide, nothing really happens. But then when we incubated it with a target RNA, we saw that CSX30 was neatly cut into 2 pieces, evidenced by this arisal of these cleavage fragments on a protein gel. And then if we use a Craspase mutant variant, in which the cysteine and the histidine residues are mutated to alanines, this effect was abrogated, independent of whether a target RNA or non-target RNA was present. So this really pinpointed that these 2 residues are responsible for that proteolytic activity. And because we had these nice detailed structures from the cryo-EM experiments, we can also say something on how this is mechanistically orchestrated at the angstrom level. So here you see Craspase, and for visibility I left out TPR, the GRAMP, which would be sitting here. Then here in green and yellow is TPR chat, and this is the guide RNA. So if you now start the video, then you see that a target RNA will bind to the guide RNA, and there will be a steric clash here with the tip of the target RNA with an alpha helix in TPR chat. And this steric clash will relay a conformational change into the proteolytic pocket where these cysteine and histidine residues are located, leading to this large rearrangements of these amino acids. To me, it always looks like a bit of a stellar constellation or something like that. And this movement, although we are still working out the exact biochemical model, we think it works something like this, that in the inactive Craspase state, so when no target RNA is present, the histidine and cysteine residues are quite distantly apart, preventing chemistry from happening. But then the moment the target RNA binds and this rearrangement takes place, that the cysteine and the histidine get closer to one another, to the point that the histidine can deproteinate the cysteine, and the cysteine can then instigate a nucleophilic attack on the CSX30 backbone to cleave it in 2 pieces. Under construction, this model, but this is how we think it works. So in empty cells, Craspase is surveying the cell, looking for complementary target RNAs. Then when a virus infects and it happens to carry the target site, upon transcription, Craspase will be able to recognize that and then switches on the protease. And then upon cleavage of the target RNA, the Craspase protease is indeed switched off again. It can then release the target RNA such that it goes back to an empty state, and then it can recycle for a new recognition event. Now you might be wondering, like, how is cleavage of CSX30 actually leading to a viral immune effect? So this has recently been shed some light on by different research groups. So it was found that CSX30 here in purple is actually in a complex with 2 other proteins as well. One of them being a transcription factor. So in the bound state, the transcription factor is in an inactive state, but then the moment CSX30 is cleaved, the transcription factor is liberated, such that it can start to transcribe genes in the bacterial chromosome. The proteins that are arising are unknown at this point, so that will be figured out. But what was also seen is that cleavage of CSX30 leads to cell death in these cells. And the thinking here really is that committing suicide, once you're infected, takes down the invading virus as well. And in this way, you prevent spreading to neighboring cells. This is kind of a akin selection strategy. So many open questions still, but there's now various groups looking into this. So I think very soon we'll know more details. So finally, I'd like to thank the people that are involved in this research. So people from our lab, the Stan Bruns lab, then Chungi and Ailong from Cornell University, who did the cryo-electron microscopy work, and then also one non-human collaborator, E. coli BL21AI, who did basically most of the work in this project. I'd also like to thank you for your attention, and if there's any questions, I'm happy to take them if time allows for it, and otherwise we can have a chat after the meeting. So thank you very, very much.